Hi everybody, this is Introduction to Biological Anthropology, Anthropology 225 Online. I'm Sharon Gursky-Doyen, your professor for the semester, and the title of today's lecture is Mendelian Inheritance. Very surprisingly, Darwin knew next to nothing about inheritance. He believed that if he mated a person who had blue eyes and with someone who had brown eyes, you would get gray eyes. Similarly, if you had someone with green eyes and they had brown eyes, they'd get a combination olivey eye, and so on and so forth. The problem is that if you think about it for a second, if everything was a mixture, one half of the variation would be lost every generation. Eventually, everyone's going to have exactly the same color eyes. If Darwin's blending theory were in fact accurate, new advantageous variation would be lost every generation through the process of dilution. Half the variation would be lost every generation. We know that that is not the case, so we know that Darwin's theory of inheritance is not accurate. The true nature of inheritance was not really known until the turn of the century with the rediscovery of the work of a Slovakian monk named Gregor Mendel. Gregor Mendel attended the university, and while at the university he was exposed to a new theory called cell theory. The basis of cell theory is that all organisms are composed of cells, and cells are the fundamental unit of all living things. Cell theory raised many questions in Mendel's mind and minds of many other scientists, including whether or not both parents contributed equally to offspring when they were produced. And it was in 1854 that Mendel began a series of experiments that began to help resolve this question. Basically, he wanted to understand how traits are inherited from generation to generation. In order to do this, he did a variety of experiments with pea plants. The first thing that Mendel did in his experiments was that he tried to identify plants that bred true. And by that I mean each parent only produces one kind of offspring in all future generations when self-crossed. A cross refers to a mating or a reproduction between two peas. Pea plants are unusual in that they have both male and female parts, and so they can self-fertilize. So he took purebred plants, pea plants, for example, that were all yellow and always produced yellow offspring. Pea plants that always were green and always produced green offspring. These he referred to as purebreds. These purebred plants are referred to as the P1 generation. It took him over 2 years to establish purebred plants. The second step that Mendel did was he selected 7 characteristics of the plants, 7 traits, which had 2 distinct phenotypes. A phenotype is the observable expression of the trait. So he looked at things like seed shape. It was either smooth or it was wrinkled. He looked at seed color. It was yellow or green. 2 distinct phenotypes. He looked at pod shape. It was either inflated or conflated. Pod color, green, yellow. Flower color, purple, white. Okay, so these were 2 distinct phenotypes. Phenotype 1, phenotype 2. Some of the phenotypes were dominant, some were recessive. A purebred plant, which always produced yellow seeds, would always produce offspring with yellow seeds when crossed with another yellow seed plant. Over the next 8 years, Mendel looked at over 28,000 plants. So this was not something that just happened overnight. It was something that took quite a long time for Mendel to do. Mendel's first experiment was monohybrid crosses. The parent individuals varied from one another in only one trait. So a monohybrid cross is a mating between 2 individuals who differ with respect to a single characteristic. So whether they differ in terms of whether the parents are yellow or green, or whether the parents are smooth or wrinkled, or whether the parents have purple flowers or white flowers. All of these, the parents differed in one characteristic. Mendel then produced hybrids. A hybrid is the offspring of a monohybrid cross, and they are referred to the letter F and with a generation number. So the offspring of a P1 with yellow seeds and a P1 with green seeds is an F1. He found that all of the F1 plants had the same trait, yellow seeds. The traits that prevailed, he called dominant. The traits that disappeared, he referred to as recessive. So this is just an example. He took a P1 green and a P1 green, and in the second generation always produced P2s, greens. P1 and P1 always produced yellow. With a monohybrid cross, a parent green and a parent yellow produce offspring that are yellow. Okay. The next set of experiments that Mendel conducted involved two hybrid plants, two F1 plants. He found that the recessive traits reappeared in a ratio of 3 dominant to 1 recessive. The third set of experiments involved the F2 generation, the F2 greens. He noted that when self-crossing the F2 greens, they only produced green seeds, but the F2 yellows produced green and yellow in a ratio of 3 to 1. So some of the crosses produced all yellow seeds, others produced 3 plants with yellow seeds to every 1 green seed. So what could explain these results? These results of Mendel's suggested 2 very important facts. The first is the law of segregation. First, it appeared the various expressions of a trait were controlled by discrete units that occur in pairs, and offspring inherit 1 unit of each pair from each parent. This involves Mendel's first law of inheritance, Mendel's law of segregation. Today, we know that meiosis explains Mendel's law of segregation. Each of Mendel's 7 traits is controlled by a pair of genes in the plant, 1 on each chromosome in a pair. These genes take different forms called alleles. During the reproductive cycle, the chromosomes separate from one another so that each gamete has only 1 gene, 1 allele for each trait. And after fertilization, the 2-gene state is restored. The second major finding of Mendel's study is that the color, which was absent in the F1 generation, had not actually disappeared at all. It was masked, and it could not be expressed, but it was still present. Masking variation in the law of segregation and what maintain variation, so natural selection has variation to act on. A phenotype is an observable physical feature of an organism. You can have yellow seeds, you can have green seeds, you can have smooth seeds, you can have wrinkled seeds. The genotype, however, is the genetic makeup of an individual. Each individual, as mentioned above, has 2 copies of each gene, 1 from each parent. These copies are referred to as alleles. Different alleles of the same gene influence the phenotype of an individual so that different variations of a trait are expressed. Dominant alleles are referred to with capitals, recessives are referred to in lowercase. So we would refer to the allele for green seeds as lowercase g and the allele for yellow seeds as uppercase Y. Individuals with 2 copies of the same allele are referred to as homozygous. Individuals with 2 different alleles for a gene are referred to as heterozygous. These individuals express the dominant allele while the recessive allele is masked. Since each individual has 2 alleles for each gene, there are 3 possible combinations of alleles or genotypes for Mendel's P's. We can have YY, homozygous dominant genotype with a yellow phenotype. You can have GG, the homozygous recessive genotype or a green phenotype. And you can have YGGY, the heterozygous genotype, which has a yellow phenotype. The crosses between pea plants are often described using what's referred to as a Punnett square diagram. This square represents the different ways in which the alleles can be combined during any specific cross, all the possible genotypes and phenotypes. In this way, the figure demonstrates the genotypes that are possible. It just demonstrates which ones are homozygous recessive, it demonstrates which ones are homozygous dominant, and it demonstrates which genotypes are heterozygous. Punnett squares can also be used to predict the proportion of genotypes. Each parent is contributing one allele at each locus, so there's a 50% chance of transmission for any given allele by a parent. So if both parents have the genotype YG, then each parent has a 50% probability of passing on the Y allele and a 50% probability of passing on a lowercase g allele. Given these probabilities, we expect 25% of the offspring to have the genotype homozygous dominant Y, 50% to have the heterozygous YG genotype, and 25% to have the homozygous recessive GG genotype. It's very important to note that Punnett squares only give expected probabilities. The different distributions may occur because each offspring is an independent event. So there's a 50% chance that any child will be male or female, but a couple may still end up with 4 girls because the genotype of one child does not influence the genotype of the next. Parents with recessive alleles can sometimes produce children with different phenotypes than either of the parents. That is, dominant alleles sometimes mask recessive alleles, and this is why a person with a heterozygous genotype shows the dominant genotype. Consider the following example. We have 2 parents with the genotype TT at the PTC tasting locus. Both parents have the taster phenotype, the ability to taste. PTC is dominant. So what genotype and phenotypes are the children likely to have? Well, 25% will be homozygous dominant for the taster, 50% will be heterozygous for the taster, and 25% will be homozygous recessive for the non-taster. Phenotypically, 75% will be tasters and 25% will not be tasters. Even though both parents have the taster phenotype, in this example, 25% of the offspring are going to have a different phenotype than the parent because of masking variation. All of Mendel's experiments and examples so far have involved what are called monohybrid crosses. After Mendel established his first law of inheritance, he extended his studies to more complicated situations and began to conduct experiments. Instead of involving just one set of characters, but 2 sets of characters. He referred to these as dihybrid crosses. A dihybrid cross is a cross between individuals who differ with respect to 2 gene pairs. For example, parent 1 not only has yellow seeds, but smooth yellow seeds, so 2 aspects, 2 characters. Parent 2, in addition to having green seeds, is they're always wrinkled. They're the same in all other 5 traits. The thing you need to remember is that yellow is dominant, as is smooth, and green is recessive, and wrinkled is recessive. So what they did, what Mendel did, was he made it yellow smooth purebred with a green wrinkled purebred. Because yellow and smooth are the dominant traits, we expect a cross between a yellow smooth plant and a green wrinkled plant to produce all yellow smooth plants. And not surprisingly, that's exactly what happens. Mendel then crossed 2 yellow smooth plants from the F1 generation, and this produced slightly more interesting results. When you crossed an F1 yellow smooth with an F1 yellow smooth, 9 out of 16 were yellow smooth, 3 out of 16 were yellow wrinkled, 3 out of 16 were green smooth, and 1 out of 16 were green wrinkled. Why did this happen in this relationship? It suggests that each of the characteristics, whether you're smooth or whether you're wrinkled, whether you're yellow or whether you're green, segregates independently. The distribution of traits in the previous example demonstrates that there is no relationship between the two traits. The expression and segregation of one trait is not influenced by the expression of the other trait. Characters segregate independent of one another. There is nothing to dictate that smooth plants must be yellow. There's nothing to dictate that wrinkled plants must be green. The expression and segregation of one trait is not influenced by the expression of the other trait. And the allele for peak texture has an equal probability of ending up in a zygote that is green or a zygote that is yellow. This is Mendel's law of independent assortment, Mendel's second law of inheritance. The alleles that code for different traits sort independent of each other during gamete formation and recombine in offspring. We know that sexual reproduction shuffles genes that affect different traits and therefore produces new combinations of traits. This is referred to as recombination, and recombination is integral to maintaining variation. The units, the alleles that code for different traits, they assort independent of one another during gamete formation, and then they recombine in offspring. As with monohybrid crosses, Punnett squares can be used to illustrate the different possible outcomes of a dihybrid cross, but because of independent assortment, there are exponentially more possibilities with a dihybrid cross. There are 16 instead of 14, instead of 16 instead of 4. Think about the P1 yellow smooth, P1 green wrinkled cross described previously. Parent 1 has a genotype of YY for yellow, SS for smooth. Parent 2 has a genotype of GG and W for green and WW for wrinkled. For each parent, there are going to be 4 possible contributions of traits and alleles. Parent 1 can have Y, can have another Y, can have for trait 2, can have this S or this S. Parent 2, small g, small w, small w. Now we've combined them when they mate. We now have parent 1 could be yellow and smooth. Parent 1 could be yellow, smooth. Parent 1 could be yellow smooth or yellow smooth. Parent 2 could be GW, 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 GW. Okay, as illustrated right here. Each of these possible contributions is listed for each parent, with parent 1 across the top of the square and parent 2 on the left. Parent 1 with yellow smooth, parent 2 green wrinkled. In the first cross, notice that all possible offspring are YGSW, yellow green, smooth wrinkled. Phenotypically, they are all yellow smooth, but genetically, they are all YGSW. They're all hybrids. The importance of independent assortment is more clear when we look at the first hybrid cross, the F1 times F1 cross. Okay, we have parent 1, YGSW. This is from the previous Punnett square. Same thing. Every parent has the same exact genotype. We then look at the different possible combinations. YS, put it here. YW, put it here. GS, put it here. GW, put it here. YS, YW, GS, GW. And then we have our 4 possibilities for parent 1, 4 possibilities for the gametes for parent 2, and these are the 16 possible gametic combinations with parent 1 and parent 2. YS gives you all yellow smooth, yellow smooth, yellow smooth, yellow smooth, yellow smooth, yellow wrinkled, yellow smooth, yellow wrinkled, yellow smooth, yellow smooth, green smooth, green smooth, yellow smooth, yellow wrinkled, green smooth, green wrinkled. So you see in this example we have 1, 2, 3, 4, 5, 6, 7, 8, 9 individuals that are yellow smooth. 1, 2, 3 that are yellow wrinkled, 1 that is green wrinkled, and 3 that are green smooth. This is a 9-3-3-1 ratio of dominant to recessive alleles. Carl Pearson in the 1900s developed the Pearson's chi-square test, and he developed this for use with categorical variables. And he designed this test to determine whether the observed distribution of individuals in phenotypic categories is as predicted by chance. And he noted that it can be used to see whether experimental results of a cross are statistically different than the predicted results, much like do the traits follow Mendelian laws. And so this chi-square test is very useful in ascertaining whether Mendel's tests are following basic laws. To perform this chi-square test, the first thing we do is we record the observed number of organisms in each phenotypic class. How many green peas? How many yellow peas? We calculate the expected values for each phenotypic class based on the expected ratios. Lastly, or thirdly, we calculate the difference, the D, in each phenotypic class by subtraction. Observed minus expected equals D, difference. Fourthly, for each phenotypic class, we square the difference, square D, and divide by E, divide by the expected ratio for each class. We add all these numbers in step 4, and you get what's called the chi-square value. If there's no difference between observed and expected values, chi-square will be 0. The value will increase as observed versus expected increases. To interpret your chi-square value, you need to convert it to a probability. The first thing you do is you calculate your degrees of freedom. The number of phenotypes is 2. So your degrees of freedom is 4 minus 2 equals 2. So your degrees of freedom is 2. You consult a probability chart to find the probability of obtaining a result by chance. This is your p-value. If p equals 0.05, the observations do not fit the expected distribution and the result is significantly significant. So if the chi-square was 0, it needs to be 5.99 for a 0.05 probability with 2 degrees of freedom in order for it to be statistically significant. I want to make it very clear that early studies of inheritance revealed that Mendel's principles don't always work. Traits that are determined by genes carried on different chromosomes do in fact behave the way he described. And interestingly, some of the 7 traits that Mendel studied in pea plants are located on the same chromosome, but in his experiments demonstrating independence, by chance, he managed to choose pairs of traits that are in fact located on separate chromosomes. When alleles are on the same chromosomes, they tend to remain together in the formation of gametes. This is referred to as linkage. Linkage is when alleles are on the same chromosome and so are inherited together. Linked alleles are not inherited independently. If 2 genes are linked, theoretically, only 2 types of gametes are going to be produced instead of 4 predicted by independent assortment. This is not always the case. Independent assortment of genes on the same chromosome occurs through the process of crossing over, where segments of DNA switch between paired chromosomes during meiosis. With crossing over, there's literally a physical exchange of genetic material between homologous non-sister chromosomes. Example, two chromosome 21s from different parents may actually exchange genetic material. For example, in this picture, there are three genetic loci on the chromosome, A, B, and C. You could have the genotype dominant A, recessive B, dominant B, recessive B, dominant C, recessive C. One of your parents contributed 3 recessive alleles, A, B, C, and the other 3 dominant alleles, A, B, C. Since these genes are all on the same chromosome, they're expected to be inherited together. You get all the recessive alleles together or all the dominant alleles together. However, during meiosis, one pair of chromosome crosses over at the C locus, producing gametes with the genotypes ABC, all recessive, ABC with one dominant C allele, dominant A, dominant B with a recessive C, and ABC with all dominants. So we see that we have 4 new possibilities instead of just all recessive, all dominant. So your potential children have more potential variation than if they just inherited one of the 2 sets of the alleles that your parents gave you. I want to make it very clear that crossing over does not change the genetic material. The alleles are still the same, they just are occurring in different combinations. So what's happening is crossing over increases genetic variation by providing new combinations of alleles. The further apart 2 genes are on a chromosome, the greater the chance that they will be separated by a crossover. For example, recessive A is more likely to be separated from recessive C than from B because the chromosomes can break between A and B or between B and C. And there's more places between B and C for the chromosome to break than between A and B. The rules of Mendelian inheritance, so elegantly described by Gregor Mendel for garden peas, also apply to humans. Indeed, the exact principles of segregation and independent assortment also account for the transmission of Mendelian traits in humans. A Mendelian trait is one that is influenced by a single genetic locus and follows a simple pattern of inheritance. It was in 1902 that the first Mendelian trait in humans, brachydactyly, short fingers and toes, was demonstrated. I also encourage you to see the webpage at the online Mendelian Inheritance in Man website. It's a database of 5,000 human Mendelian traits. A good example of human Mendelian trait includes the blood groups. These are reflected as protein antigen specificities on the surface of red or white blood cells. This was first discovered by Karl Landsteiner in 1924, and he hypothesized that the 3 phenotypes A, B, and O were based on 3 alleles of a single gene. The gene is I-isoagglutinogen, short for antigen, and it's located on chromosome 9. As I said, there were 3 alleles, A, B, and O. An individual can only possess 2 alleles, and those alleles code for antigens on the surfaces of your red blood cells. If only the A allele and or O is present, the phenotype is going to be A. A is dominant over O. O antigens are produced. If only B and or O are present, the phenotype is B because B is dominant over O and B antigens are produced. If both A and B are present, the phenotype is AB. A and B are both codominant, so both A and B antigens are being produced. If neither A nor B is present, the phenotype is considered O because O is recessive to both A and B. So why are there 3 alleles? Both A and B antigens are derived from a precursor molecule called the H substance. The H substance can be incompletely formed due to a recessive mutation, little h. Malformation prevents formation of either A or B and results in O. This is sometimes known as the Bombay phenotype after the first woman identified with it. Codominance is seen when two different alleles occur in heterozygous condition, but instead of one masking the other, the products of both alleles are seen in the phenotype. For example, AB blood. Alleles of A and B antigens are both present. They are codominant. Red and yellow roses. We have the alleles for red and we have the alleles for yellow, and they are codominant, and they are both expressed. Sometimes, however, we have what we call incomplete dominance. Incomplete dominance is seen when a heterozygote has a phenotype which is intermediate to those of homozygous parents. For example, pink roses. The alleles for red and white are incompletely dominant. The alleles are not blended. If two pink plants are crossed, they will produce red, pink, and white F2 plants in a 1:1 ratio because pink flowers need both red and white alleles. Because humans cannot be used in experimental breeding programs as were Mendel's peas, a more indirect approach must be used to demonstrate patterns of inheritance. The principal technique that is used in human genetic studies requires the reconstruction of pedigrees. Pedigrees are charts of matings that have already taken place and resulting offspring that have been produced, and they are generally shown in human families over the span of a few generations. Pedigrees help determine if a trait is Mendelian. They also help you identify the mode of inheritance. Pedigrees traditionally use a set of standardized symbols, many of which are borrowed from genealogy. In constructing a pedigree, males are represented by boxes and females are represented by circles. An individual who exhibits a trait has a filled square or a filled circle. Heterozygote carriers, when known, have half-filled circles or squares. The relationship between individuals in a pedigree are traditionally indicated by a series of lines. A horizontal line between individuals at the middle of the square box indicates a mating. Brothers and sisters are connected by horizontal lines at the top of their markers. Children are connected to parents by vertical lines. Generations are generally indicated by Roman numerals, and individuals within each generation are generally indicated with Arabic numerals. This is an example of a pedigree. As you can see right here, this is generation 1, 2, 3. These are the grandparents, these are their children, and these are the grandchildren. This is the female, grandmother, grandfather. Within the first generation, they had 3 children. This son, number 3, this daughter, number 5, this daughter, number 7. This son, number 3, married a woman. Daughter number 5 married a man. Daughter number 7 married a man. 3 and 4 had 2 children, a daughter and a son. 5 and 6 had a daughter and a son. 7 and 8 had a son and a daughter. The individuals with the filled ones are individuals that exhibit the trait. We can then use this pedigree to determine the patterns of inheritance for that specific trait. In general, there are 6 primary modes of Mendelian inheritance that have been described in humans. These are autosomal dominant traits. These are autosomal recessive traits. We have Y-linked, X-linked dominant, X-linked recessive, and mitochondrial. And I'm going to go through each of these individually. We start out with autosomal dominant traits. These are ones that are located on your non-sex chromosomes. So any chromosome that is not affiliated with determining whether you're male or female. As I noted, they're present equally in males and females. because they're not associated with your sex chromosomes. Classic examples of autosomal dominant traits are things like brachydactyly, Huntington disease, and PTC tasting. With autosomal dominant traits, we generally see that 50% of the offspring are expected to show the trait when only one parent is affected. 75% of offspring will show the trait when both parents are heterozygous. This is due to the principle of segregation, which we already discussed, and the process of meiosis, which we also discussed. Counting the number of individuals of each sex in the pedigree will show that the disease does not affect males or females more frequently than the other. Once again, it's because it's an autosomal dominant trait and it doesn't skip generations. Autosomal recessive traits are also located on non-sex chromosomes. They are equally present in males and females, but they differ than autosomal dominant traits in that they skip generations. So it creates carriers, individuals that are heterozygous for a condition. Classic examples of these autosomal recessive traits in humans include medical conditions like albinism, cystic fibrosis, PKU, sickle cell anemia, and Tay-Sachs disease, in addition to a variety of others. And once again, if you check the online Mendelian Inheritance in Man webpage, you can find a whole bunch more of these conditions. We then have X-linked dominant traits. These are located on your female sex chromosomes, so they're only located on your X chromosomes. But males do have X chromosomes, so they can be found in males. But because females have twice as many X chromosomes as males do, females are affected twice as often as males. Classic example of an autosomal, an X-linked dominant trait is something known as hypophosphatemia. This is known as bow-leggedness and rickets. We also have X-linked recessive traits. These are located on your female sex chromosome. Once again, males are affected more than females. Why do we see males affected more than females with X-linked recessive, but females affected twice as often with X-linked dominant? Because when a male receives an X-linked recessive trait, it is always expressed because males only have one X chromosome. Examples of conditions where males are much more likely to express these, twice as much more, are things like hemophilia and red-green color blindness. We also have what's known as Y-linked traits. Y-linked traits are located on male sex chromosomes. They are only present in males, and classic things for Y-linked traits are things like testes determining factor. It's also been suggested that an X-linked trait that is found in humans is hairy ears. You'll note that you only see, old men with hairy ears. You rarely, if ever, will see an old female with hairy ears. And once again, it's been suggested that it is linked to those Y chromosomes. So with an autosomal dominant trait, we note that 50% of offspring are expected to show the trait when one parent is affected. Let's take a look at this. So this parent, this female, is affected. This father is not. We know it's affected, one, because it's colored in dark, and two, it's a dominant trait, and we give it a capital letter. So these two parents are giving this son this genotype, this daughter this genotype, and this daughter this genotype. 50% of offspring, if you do a Punnett square between these two traits, you'll see 50% of the offspring, on average, will have this condition. 75% of the offspring will show the trait when both parents are affected. So if you do another Punnett square and you have both the female and the male with this genotype, you're going to see 75% of offspring are going to have it. You'll also note that in autosomal dominant traits, there is no sex bias. That is, males and females are equally likely to express the trait. There's really no difference. You also see that it doesn't skip generations. It's present in each generation. These are classic for X-linked dominant traits. for autosomal dominant traits. This is an example of of pedigree for an autosomal recessive trait. Now remember, with a recessive trait, we can have carriers and you can have individuals that are exhibiting the trait. In this example, you have an individual, the father, who exhibits the trait, and he has 2 recessive alleles. This mother has a recessive allele and a dominant allele, so she's a carrier. And we know that she must be a carrier because this child here, this daughter, has 2 recessive alleles. We know it must have gotten 1 recessive allele from dad, but where'd they get the second one? It had to come from mom. Since son 3 and daughter 7 don't exhibit the trait, she has to be heterozygous. One of the things you see with autosomal recessive, once again, is that there's no sex bias. It's equal likely probability that both the males and the females will exhibit the trait. It does skip generations, though. It's not really clear in this pedigree, but what you will see is that it does skip generations. And it produces a lot of a lot of carriers, a lot of individuals who have 1 recessive allele but don't exhibit the condition, and that's what we're seeing here. So this individual, and this individual are carriers, and then they pass it on to their children. This one passed it on to his child even though he didn't have it. The proportion of affected phenotypes is usually 1 in 4. So if you would do a pedigree, a Punnett square of these 2 individuals, the probability that their offspring would actually have this autosomal recessive condition is 1 in 4. As I mentioned, in addition to having Mendelian inheritance in your autosomes, we can also have Mendelian inheritance patterns in terms of our sex chromosomes. Sex-linked traits are found on your sex chromosomes. Almost every one of them is found on the X chromosome. This is an important exception to the rule that individuals contain 2 alleles of every gene, because females have 2 X chromosomes. Males only have 1 X and 1 Y, so they don't have an actual pair. We refer to this as hemizygous. So males are hemizygous for genes carried on the X chromosome. They're not hetero and they're not homozygous. For X-linked genes with 2 alleles, you're always going to have 3 female genotypes: homozygous dominant, heterozygous, and homozygous recessive. But for the male genotype, you're only going to have 2 genotypes: dominant and recessive. For Y-linked genes, once again, there are only 2 male genotypes, dominant and recessive, and there will be 0 female genotypes. So very, very different pattern than what we see with the autosomes. This is an example of a pedigree of an X-linked dominant trait. So if they have it on their X chromosome, it's going to be expressed. So what we see is that X-linked dominant traits are actually quite rare. There aren't very many of them. We see that in this example we have an X-linked dominant, and this is the male. He has a recessive and a Y. They pass it on here to this son in the heterozygous condition, to this daughter in the heterozygous condition. This son then passes it on to this daughter in the heterozygous condition. Now remember, it's getting passed on in the heterozygous condition a lot because it is an X-linked dominant trait, so you only need 1 allele in order for it to be expressed. What you see is that you're more often going to see it in males because they only need 1 trait, but you're not going to see you're going to see affected males produce affected daughters, but they're not producing affected sons because these males are always passing on their Y chromosome, not their affected X, to their sons. So you'll see twice as many females affected as you do males in terms of X-linked dominant traits. With X-linked recessive traits, you're not going to see any male carriers. Once again, why? Because the fathers are only passing on their Y chromosome, okay? So they're not passing on their X, and so you can't get it in that manner. We'll see males are affected more than females, So what we see is, as an example, with color blindness, 7% of European men have red-green color blindness, while less than 1%, or less than half of 1%, of European women have red-green color blindness. So males are affected very substantially higher rates than females. I will point out that the red and green the alleles that allow you to see red and green are located side by side on the same chromosome, and that's one of the reasons they're often inherited together. And then lastly, the last mode of Mendelian inheritance that we see in humans is Y-linked. At present, there are only about 12 traits that are known to be inherited on the Y chromosome. Obviously, they only appear in males. They are always inherited from the father. There's some interesting features of the Y chromosome. First of all, it's minuscule. Remember I mentioned it was minuscule. And there are regions of the Y which are homologous to the X, so they're exactly like the X. But what we see is that the genes on the Y, even though they look exactly like those that are on the X, are actually completely non-functional. And it's been suggested by some that one, the Y chromosome is not essential for viability, and two, that the X chromosome developed into a Y. So just an interesting question about whether or not Ys are really needed. But as you see, there are only 12 of them, and some of them are very important. As mentioned, testes determining factor, very important for males to become males. And of course, hairy ears, another very important trait. Because Y chromosomes are only and always inherited from the father, they have been used sometimes to trace male parentage. As a classic example, Thomas Jefferson had a son with his slave, Sally Hemings, and the parentage of his son was tested by comparing Jefferson's brother's son's DNA to that of Eston Hemings' son's DNA, and it was found that this truly was his son. It was a very, very high probability that it actually was his son. Another example of how studies of the Y chromosome have been used is in terms of the cone modal haplotype. The cone haplotype is shared by many Jewish priests, and it's thought to have been shared by the biblical Aaron because the Jewish priesthood is patrilineal. Problem is, it's also been found in groups of non-Jews, so it hasn't been as useful a technique for understanding religion as some would like. Now, so far, when we've been talking about Mendel and Mendelian inheritance, we've really only been looking at single traits that have been determined primarily by a single pair of genes. Most of the time, it's not that simple. A single gene often has more than one effect. It can have multiple effects. This is known as pleiotropic effect. Multiple genes can also be required to produce a single effect. This is known as polygenic, many genes. Polygenic traits are also known as quantitative characters as well as continuous characters. So there are several signs to indicate whether or not a trait is quantitative. The first is that it can be measured. If it's something that can be measured like height, then a trait is quantitative. If it has a wide range and can be affected by environmental variables, then it is also something that is considered quantitative. And lastly, a sign that a trait is quantitative is that the offspring of individuals at the extremes will often have a less extreme phenotype. A special case of variation in expression includes genes that are expressed differently in both males and in females. Male pattern baldness, in which hair loss occurs prematurely from the front and the top of the head, but not the sides, is a classic example of a pleiotropic effect, where you have pleiotropic genes that are operating. Male pattern baldness, as you know, probably predominantly affects males. Females with the gene show thinning of hair. And it is believed that male hormones such as androgens are involved in the expression of this gene. Males whose testes are injured or are castrated do not lose their hair, but they will if they are given hormone treatments. Females who have adrenal tumors that cause them to secrete large amounts of androgen also lose their hair. This suggests that the gene for pattern baldness is dominant in males, and in females it is recessive. We also have what are called polygenic traits. Polygenic traits are traits that are affected by many genes, but they can also be explained by Mendelian inheritance. Many genes, each of which is controlled by a number of alleles, each contribute to the trait, and these traits exhibit a continuous distribution. Each gene follows Mendelian patterns, but the trait overall follows a continuous pattern. This distribution of these traits follows a normal bell-shaped curve, and most people have phenotypes between the extremes. This is known as quantitative inheritance, and it's very different than discrete characters like pea color, where it's either green or it's yellow. It's quite different. The fact that most offspring of individuals at the extremes of a polygenic distribution are less extreme than their parents is called regression to the mean. And this is a distinguishing characteristic of most polygenic traits. Offspring of individuals with a phenotype from the extremes are generally have a less extreme phenotype. A classic example was a study done by King Frederick William. He wanted to conduct a bunch of giant guards. He wanted to create giants to act as his guards. This was in Prussia between 1675 and 1740. So basically it was an attempt at a human breeding program, and these are hypothetical genotypes. So what he tried to do was, what would happen if, one of the questions he was trying to ask was, what would happen if a 6'9" male mated with a 6'3" woman? And these are the hypothetical genotypes. So if we start out with a 6'3", which is the shortest one we have, okay, and for each time they add 3 inches, we put a capital. So this would be 6'6", 6'3", 6'6", 6'9". So we start out with a 6, okay? And so this would be a basic of an individual who is at least 5'9" or 6 feet tall. And then you add 3 inches for each capital. So 5'9", so 6, 6'3", 6'6", 6'9", 7, 7'3". Okay? So this individual has to be at least 5'9" inches tall with this genotype. And this is the example. And these are the alleles from the mother, father. Remember, you're going to get 3 alleles from mom, 3 alleles from dad, with 3 different factors, because we're looking at polygenic. And you'll see that these are the different gametic combinations from mom and dad that are possible when you combine each of these factors. And then you create a pedigree of all of them. And you have a 6'9" male, right? And these are the possible gametic combinations, and a 6'3" female, and these are the gametic combinations, and then we combine them together. And one of the things that we see as we go through here, and we use this color code, that the majority of the offspring are shorter than the parents. So we don't have anybody who's gray, but we do have some individuals that are this light brown. So these 4 potential offspring all have phenotypes shorter than the parents. These oranges here are 6'3", so they're as short as the mother. Okay, these 6'6" are the purples. Okay, these oranges are the 6'9", and these are 7 feet. So there are only 4 possible offspring out of all these options, 4 gametic combinations that actually produce offspring greater than the father. Okay, the majority of them are somewhere in the middle, 6'6", right in the middle. This is regression to the mean, characteristic of extreme phenotypes. So most of the children have heights that are intermediate to their parents, showing regression to the mean. 68.75% of the combinations resulted in children shorter than their father. In succeeding generations, further regression to the mean continues to occur. Now, heritability is a measure of the proportion of the total observed variation that can be contributed to genetics rather than the environment. It is a very important statistical concept. A classic example where people have been very interested in studying heritability, of course, is IQ scores. It's well-known and controversial use for heritability statistics. There's a normally distributed continuous variation in IQ scores. Most people accept that both genetics and the environment play a role in IQ score performance. They become very distraught when we discuss the issue at ethnic levels, and I'll talk a lot more about this when we discuss human variation. One of the things that is noted in terms of these ethnic differences is that it's been noted that Asian Americans score best on IQ tests, followed by European Americans and then African Americans. It's very important for you to recognize that these IQ differences do not result from genetic differences. Heritability scores apply only within a population, not between a population. So anytime you read anything that says that African Americans are genetically inferior intellectually, please know that that is an inaccurate application of statistics. Or if they were to say that Asian Americans' IQ scores are higher than European Americans because of genetic superiority, once again, that is an inaccurate application of these statistics for heritability. Now, one can observe natural selection working on continuous characters in a variety of ways. We can have different types of selection. We can have stabilizing selection, we can have directional selection, and we can have disruptive selection. One of the main types of selection that I just mentioned is called stabilizing selection. Stabilizing selection effectively selects out the extremes. Okay, there was this guy, Herman Bumpus, and he was interested in studying bird wings. And he looked at all the birds that died in a big storm, and he found that they tended to have wingspans of longer or shorter than the average wingspan in the population. In this example, the birds that died had longer or shorter wings than average. Natural selection was operating, and it was selecting against the extremes, against the really long wings, against the really short wings. Natural selection, when stabilizing selection is operating, is acting against any change in form. It wants to maintain the status quo. Another classic example of stabilizing selection occurs with human birth weight. The weight of a newborn infant is the result of a number of environmental factors, such as the mother's weight, her age, history of smoking, and there's also a genetic component to infant birth weight. Newborns who are very, very small, usually less than 5 pounds, are much less likely to survive than are newborns that are heavier. Very small babies are more prone to disease, they have weaker immune systems, and thus their survival is much more difficult. Newborns that are very large are also likely to be selected against. And this is because a large infant creates complications during childbirth, and both the mother and the infant die. So what we've seen over time is that there is selection against both extremes. Selections against very small babies, and selections against very large babies. So there's a real very tight relationship between infant birth weight and infant mortality. Okay. And then we have what is known as directional selection. With directional selection, selection is acting against one of the extremes. Okay, so here we have stabilizing selection, and it is getting rid of all of those on the high end of the distribution and the low end of the distribution. With directional selection, it is literally pushing the mean over. So the mean is no longer here. The mean, the average, the median has moved over here. So, and a classic example of directional selection concerns salmon. It was in 1954 that fishermen began to be paid instead of by individual, but by the weight. I think I said that backwards. They started to be paid by weight instead of by the number of individual salmon that they were capturing. So as a result, they began to increase net use. And because these gill nets selectively harvest larger fish. And when you're paid by weight, the bigger the fish, the more you're going to get paid. The selection for small size among the salmon was very intense because 75% of the salmon were being caught in the gill nets. The only ones that weren't being caught in the gill nets were the really small ones. So there was intense selection for small size. Only those that were small enough not to be caught in the nets survived. So what they found was that the average weight of the salmon decreased by 1/3, by 33%, over 25 years. The last way that natural selection operates on these continuous characteristics is what is called disruptive selection. Disruptive selection selects against the mean. So instead of selecting for the mean, which is what's happening with stabilizing, or moving the mean, which is what happens with directional, it's selecting against this. So it basically is creating a bimodal distribution. What you have here is natural selection is favoring the really small ones and the really large ones, but not the median ones. In nature, it has been suggested that sexual dimorphism, the difference between males and females in body size, is a result of disruptive selection.